I'm surprised how many people are left to see some R code. I'm very impressed by that. So I will now talk about how we analyze gene expression data with R and the WGCNA R package. So because I'm on video, let me introduce myself. I'm Steve Horvath from the University of California, Los Angeles. And I will now describe where you can find this tutorial, the R software tutorial, and the related data. If I had a lot of time, I would help you download the data, and then we would go through the commands one by one. But I think only few of you will be able to be fast enough to follow along, but maybe some of you can do it. In any event, let me start with a Google search. Let's say I want to learn about WGCNA, and I just put in weighted network into Google. And there's some entries, and one of them, the second hit, is welcome to the weighted gene co-expression network page. So we click on it, and let me briefly tell you how this web page is organized. So one thing you see is there are PowerPoint slides that provide an overview of WGCNA and so on. And then there is a link to a book, and we will go there shortly in order to download the software tutorial. But also there are now links to the workshop. And I'm afraid these links are not yet updated, but this is where you will find all of the PowerPoint slides from the speakers and links to the videos, There are also links to old workshops, but you get a sense, The next thing I want to mention is WGCNA is, of course, an R package, and you can install it from within R, but alternatively, you could go to this link to get installation instructions. Now, the other thing I want to mention is that there are, of course, a host of methodological papers, we call them theory papers, that describe various aspects of network construction, module detection, network statistics, and so on. So the webpage starts out with the theory papers. Often papers are accompanied by PowerPoint slides. And also when you click on one of these links, then you can download tutorials that are relevant for this particular paper. And often we post the microarray data or the gene expression data. My goal is always to publish the data and the software code that are used for generating the figures and tables of a paper to ensure full reproducibility, but also to help people learn these methods. So in any event, there's a host of theory papers. For example, Peter Langfelder's paper on how we do branch cutting, and Peter Langfelder and Bin Zhang worked on that. There are papers on how we carry out module preservation analysis. And so, for example, here, we could click on this link on tutorials, and you will now find a whole set of tutorials about how to carry out module preservation analysis. Going back, let's proceed. And at the bottom of this web page, there are applied papers, because some of you are biologists and that's your main interest. And it starts out with applications in brain cancer, and here again would be data, yeast. And the papers are arranged by time of publication. So the most recent papers can be found at the bottom. There's material from Andy Levine where we studied HIV. There are papers from songbirds, papers describing applications in autism or the Allen Brain Institute data, and so on. But in any event, let's now come to the main topic of this lecture, which is the software tutorial. So I start out here, and there's a link to this book by Weighted Network Analysis. And so we click on this link. Why? Because there is a chapter in that book, which is chapter 12, that describes these R commands. And also, you notice there's a link to the mouse expression data. So these are the mouse data from Jake Lucis's lab. And so if you wanted to follow this tutorial, what you would do is you would save these zip data into a directory on your computer. And similarly, you would download the tutorial either as a Word doc file or as a PDF file. I always use the Word document. There are two tutorials. One is the corrected tutorial. And in a second, I explain to you what was corrected. But let me just say, at some point, I removed the wrong mouse, you know. So let's focus on the corrected tutorial. So in any event, this is now the corrected R software tutorial. And it is, as I mentioned, it corresponds to a chapter in this book. You don't need to read the book in order to apply the R code. It's actually very self-contained. In any event, after some blah blah describing what we do, we we come to material colored in blue, which really reports R commands. You know? And so let me mention there is a link or a path that describes where this material is located on my computer, meaning where the gene expression data are located. Remember how you downloaded the zip data file? And so the way you interact, of course, with R is, first of all, R is a free software, and we and many others really like it. And so after opening this software, what you do is you copy and paste this blue code. That's how the tutorial works, you know. You copy and paste it, and you hope for some good output. So what do I learn here? I hope you can see it, but here we analyze female mouse liver tissues from data generated by Jake Lucis's lab, and we focus on 3,600 genes that were selected in some shape or form. Of course, there is no limitation on the number of genes you can analyze. Our group routinely analyzes, for example, DNA methylation data from Illumina, which are 480,000 variables. You know, so you can apply these tools to very large data sets. But in any event, let's just stick with this example. The next thing I will do is I will look at the names, the column names of the data that I just downloaded. So you notice I go back and forth by copying and pasting. And so these are the names. And you notice there are several columns that describe the gene symbol, the locus link ID, and various measures on the genes or probes. But then there are columns that start with F2, for example, F22. So why is that? Because these data came from an F2 mouse intercross. And so 2 is mouse number 2, this is mouse number 3, and so on. So why are these no consecutive numbers? Well, remember, these are all the female mice. The other mice are not part of this analysis. Now, moving right along, we also download what we call trait data. So I will now copy and paste this command. Let's see, I think I must have skipped some output. I think I accidentally removed some lines. I hope you downloaded a better version. One problem with copying and pasting is that sometimes you cut it and so it's gone. But no worries, because all the output is part of the tutorial, so no harm done. So let me mention that we follow a convention, which is that we introduce a file called this terminology .expression. So that is a data frame where the columns correspond to the genes, okay? Whereas the rows are the mice. And this is an important convention in R because when we, our focus is of course to construct a network among genes and to correlate these genes. And so the genes are our variables of interest, and therefore we want to have them as columns. The next thing, of course, is we downloaded these trait data. And so what we need to do is we need to align the gene expression data with the trait data. Now, in order to do that, we use the R command match, which matches the mouse IDs from the gene expression data to the IDs from the trait data. Why do we need to do it? Because the traits are available for both male and female mice, whereas the expression data are only the female mice. So then we check whether the alignment is correct. And so we get 135 trues, meaning there are 135 mouse livers and they are all perfectly aligned with the trait data. So now, one way to read this tutorial is by looking at the output, because if you look at every single blue line, you will burn out after page 1. So start with the figures. So you notice there's a nice figure, it looks colorful. What's the meaning of it? So here we actually cluster the mice, not the genes. You notice there are these IDs that indicate these are different mice. And further, underneath the cluster tree, there are different color bands corresponding to these different physiologic traits. For example, there was mouse body weight, the mouse length, the HDL levels, glucose and insulin levels, and so on. And the coloring indicates whether one of these values takes a high value or a low value. So red means indicates a high positive value. Now, the very first color band shows an outlying status for the mice. I will show to you in a second how I define it, but let me already mention that the cluster dendrogram indicates that there's this mouse number 221 that is very far removed from all the other mice, And so, of course, one of the main uses of hierarchical clustering of samples is to identify outlying samples. And given that we have here 135 mice, we really can afford to remove one of the mice if it looks suspicious. So coming back, I mean, this is clearly a valuable plot in many analyses. How do I generate it? And that's why we go back now to look at the R code that was used to generate it. So what we do here is we actually construct a network among mice. Remember I mentioned yesterday that you can form correlation networks among samples. We call these sample networks. But also you could generate a network between mice based on the Euclidean distances between mice. And so to accomplish that, we use the WGCNA R function called adjacency. The adjacency function generates an adjacency matrix. And remember, a network is characterized by an adjacency matrix. Now, if you carefully look at this R code, you notice that I transposed the data. And why is that? Because I want to construct a network among mice, not among genes. So I transposed it. And now I said, I want a network based on the Euclidean distance. So I say network, this type is a distance-based network. Incidentally, you can use any other distance, not just the Euclidean distance. You could have the Manhattan distance, Canberra distance. Most distances you could think of are implemented in this R function. Why do I do it? Because often when people cluster samples, they use the Euclidean distance, Whereas when we cluster genes, we of course use a correlation coefficient, So now when you have an adjacency matrix, what you can do is you can define a measure of connectivity. Now, which is accomplished in this R command. Remember, the connectivity is just the row sum of the adjacencies, or the column sums equivalently. And so the connectivity measures for each mouse how similar is it to all the other mice. And mice that have a low connectivity look suspicious. Now, the connectivity, of course, it very much reflects how many mice are present in the data set, and therefore it can be a good thing to scale the connectivity. Scale is the same as what is more commonly referred to as standardization. So here the connectivity is standardized to mean zero variance 1. And so this measure z dot k is really a measure of outlyingness for every mouse. And so here I choose a threshold of -5, which for finding outlying mice, which indicates that a mouse is at least 5 standard deviations removed from the other mice when it comes to the connectivity value. And that's how I find suspicious mice. Now, as you noticed, here I color code outlyingness, and so to do that, I threshold this z dot k, and I say if the zk value is less than -5, I highlight this mouse as red. All right, the next thing that I want to highlight is that we use the R command flashclust for hierarchical clustering. Some of you are very familiar with R, and you probably think, why didn't you use hclust? Hclust is a well-known R command for hierarchical clustering. Now it turns out that the people who implemented the hclust command, they made a slight coding error, which slowed down the hierarchical clustering. And so Peter Langfelder revisited the programming of hclust, and he found this coding error and just corrected it. And so his resulting function flashclust has the identical output as hclust. It's identical in every aspect except that it's faster. Now in this example it doesn't matter, but sometimes you have very large data sets and then it matters whether you use flashclust or hclust. The other thing I mentioned, when you remember, hierarchical clustering takes as input a distance measure or a dissimilarity measure, more precisely speaking. And so here we define the dissimilarity as 1 minus the adjacency matrix, and then we use average linkage hierarchical clustering. All right. The next thing I want to mention is that we, of course, want to create this dendrogram. But we want not only, a dendrogram is another word for cluster tree. So we don't just want to produce this dendrogram, but we want to also have colors underneath the dendrogram. And to do that, we use the WGCNA function plot dendro and colors. you will notice that the WGCNA package always uses very long command names because we want to be user-friendly. So the next thing I wanted to mention is that, of course, we have these clinical traits, and we need to, which are really numeric values, and we need to turn numeric values into colors. And how do we do that? So there is a function called numbers to colors, which turns numbers to colors. And so I just want to remind you, and we apply it then to every one of these columns. And if you copy and paste the blue code, then you literally get this output. Now, looking at this output, as I mentioned, I see this outlying mouse, but other than that, I really don't see any patterning, and which tells me one thing, that none of these traits lead to a globally distinct gene expression profile. These are all complex traits. As I said, this mouse F221 is outlying, and here I present some R code that shows you how to remove this outlying mouse. And this is where I mentioned I made an error in the book. So I removed this outlying mouse, and now I'm ready for my proper co-expression network analysis. Now we want to construct a network among the genes. So you remember that when it comes to WGCNA, one of the first things you need to decide upon is which threshold you use, which soft threshold. And this is the soft threshold is used for raising the correlation matrix to this power, and this power is the soft threshold. Now, we sometimes use the scale-free topology criterion, and before I go there, let me remind you that we have this scale-free topology, and we create this type of plot where we show how the scale-free topology index, the R-square measure, depends on different powers. So, for example, if I use a power of 1 or 2, then the scale-free topology fitting index is low, meaning the network doesn't satisfy scale-free topology. Whereas if I increase the power, then the R-square measure is high, and so I start to arrive at a network that satisfies scale-free topology approximately. And the scale-free topology criterion says, pick the power where the saturation curve starts to really saturate or come down. And so here one could take a power of 6 or a power of 7. It really doesn't make much of a difference, but I just will choose the power of 7. Now, in order to create this plot, we need to run, of course, this R command. And here I use the R function pick soft threshold, which does exactly what the name says, And the output of this function is a table where for different choices of the power, here I study from 1 to 10, I get this R-square measure. And you notice for a power of 7, I get an R-square of 0.92. Now, the other thing you always want to look at is the slope. So the slope tells me whether you want a negative slope, because it indicates that hub genes are less frequent than lowly connected genes, So in any event, the slope looks fine. It's around minus 1.7. There are other values that are being outputted, but we usually ignore them. So our focus is really on these two measures. All right. So according to the scale-free topology criterion, I would now choose a power of 7. And let's now assume that I'm really lazy about the analysis, and I want to do WGCNA in pretty much one R command. And I can accomplish that using the command blockwise modules. So what's the input of it? Well, I do need to input the gene expression data, and to remind you, these are just numeric values and the columns correspond to the genes. The next thing I need to specify is what kind of correlation do I want to use? And so here I say the correlation type is the Pearson correlation, but as you remember, lately we have found that it's far better to use a robust version of the Pearson correlation, and so we would use the bi-weight mid correlation. So I would specify core type equal to bi-weight. Another thing I need to specify is the network type. And so for historical reasons, I used the unsigned network in this tutorial. And why? Because I want that the results are congruent with previous publications from 2006. But of course, if I could go back in time, I would definitely use the signed correlation network, and I would just say network type equal to signed. Finally, we need to specify, well, which power do you want? And in this case, as I said, we choose a power of 7. But in any event, let's go back and reverse things. And we just proceed with the old way of doing things. We use a Pearson correlation, an unsigned network on this power. I do want to mention that when I chose this power using my R command pick soft threshold, as the default, this command assumes that you have chosen an unsigned network. But of course, if you already know ahead of times that you will use a signed network, then you would already use this argument for here for picking this soft threshold. Do you see I can specify network type equal to signed? Most of our R commands that form correlation networks have the argument either network type or type, and then you would say signed if you want to choose a signed correlation network. In any event, so I choose these inputs. There's one other input which is called the merging thresholds. So remember that two modules will be merged if their eigengenes are highly correlated. And so the merging threshold, roughly speaking, is 1 minus that correlation. So that indicates here that modules whose eigengenes have a correlation higher than 0.75 will be merged, roughly speaking. So I copy and paste this command into R, and this will take probably a minute or so. And then what you get is the results of that are saved in an object called net. I mean, you can give it any name. For example, you could call it female liver net. And one of the first things I will be interested in is, of course, the module assignment. So the output of this is so the network results in an output which are these module labels, and the module labels are integer values. Remember, module number 1 could be the largest module, module 2 is the second largest module, and so on. And in order to turn these module labels, these integers, into colors, I now use another valuable R command called labels to colors. Okay. Actually, that's a function that not only translates integers into colors, but it also translates any categorical values. You know, you could have factor levels here. All right. The other thing that is saved in this net objects is a data frame of module eigengenes. So for each of these modules, we of course arrive at a module eigengene, and these can be accessed using that command. Remember, the net file, the net object is actually a list. And in order to access list components in R, you use the $sign. All right. So let's move on and relate things to our clinical trait of interest. So assume we want to find modules that relate to mouse body weight. And therefore, I specify here that, focus on mouse body weight in grams. And what I do here is I define a gene significance measure by correlating each gene with mouse body weight. And so I do that, of course, using the chord command. And so the gs.weight specifies this gene significant measure with respect to mouse body weight. Yes? Yes. So definitely, I often have, for example, disease status, and then case could be 1, control is 0, and you just import that, you know. And there's no reason not to report a correlation between a numeric variable and a binary variable. Things become a little bit more tricky when you want to calculate a p-value for it, but of course you can definitely also calculate a student p-value for that, a student t-test p-value for that. In my book, I have a whole section on how to turn correlations into p-values. There's a host of statistical methods for it. Apart from the student t-test, there's something called the Fisher transformation and many other methods. You know. All right. In any event, this gene significance measure is, of course, a numeric variable, but we will want to visualize it. And therefore, again, we define a vector called gene significance weight color, which whose entries correspond to colors. And to arrive at it, we use, again, numbers to colors. So let's just see what we want to create. So the R command produces this figure, which, of course, shows the hierarchical cluster tree. It shows underneath it the module assignment colors. And as a second color band, it shows the gene significance measure with respect to body weight. Now here I restricted myself to mouse body weight, but of course in practice you would want to add color bands. For example, you would want to have a color band for insulin levels. So you would then have GS insulin or GS glucose levels and so on. Why would you do it? Because it would be nice to see which modules relate to which traits. Here, brown indicates positive values, blue indicates negative values, and what we learn is there's this blue module that contains a lot of genes that have either high positive correlations with mouse body weight or high negative correlations with mouse body weight. And the reason why this module contains both positively and negatively related genes is because we use an unsigned network. If we had run this analysis with a signed network, things would look different, you know. All right, moving along. So remember how I told you earlier that we routinely apply these methods to very large data sets, for example, containing hundreds of thousands of variables. And we even do that with a laptop computer. So how do we do it? We use, of course, this blockwise modules functions, which has this advantage that it allows, that it automatically splits the data into manageable blocks. And in order to illustrate that feature, I will now assume that I have a very old computer that can only deal with 2,000 genes at a time. And so therefore I will now specify that the maximum block size that my computer can deal with is 2,000, only 2,000 genes at a time. Other than that, I leave everything the same, the network type and so on. At this point, I want to mention one thing. There's one argument in this function, which is the minimum module size. And so here I tell the function that I'm not interested in modules that contain fewer than 30 genes. This can be a valuable command if you have hundreds of thousands of variables, because when you have 500,000 variables, and you allow for very small modules, you may be flooded by modules that are not exciting to you. But in any event, so I run this analysis and save the output into an object called BWNet, meaning blockwise network. And again, I can access the resulting module assignment using the $sign. So I say BW, so this is the list component called colors, which contains the module assignment. There's one little confusing thing here, which is, although we call it colors, it actually reports the integer values, meaning the module assignment in integers, module 1, module 2, and so on. Why does it report integers and not colors? Because here I specified that it should output numeric labels. Do you see that? It says the module assignment is specified in terms of numeric labels. So in any event, the other thing I want to mention is that this is, of course, my second module detection analysis, because I had earlier carried out a module detection analysis. And in order to align things, to make it more reader-friendly, I want to reshuffle the cluster labels of the second module detection analysis so that they match the labels of the first module detection analysis. And I achieve that by using the function match labels. So notice I input now the new module assignment and the previous module assignment, and then it reshuffles the labels of the new module assignment to give me a third version of module labels that are now aligned. Okay. And so I have now these reshuffled labels from the blockwise analysis, and I turn them again into colors using labels to colors. And I can already ask what proportion of genes have the same module labels in the first and the second blockwise analysis, and I see it's roughly 70% of genes. But I can also visualize that. Now, the great advantage of the blockwise analysis is that it can deal with hundreds of thousands of variables, but this comes at a price, which is when you have small blocks, you can only visualize one dendrogram for each block, right? There's no overall dendrogram. And so here I say visualize block number 2. Meaning the 2,000 genes that are part of block number 2, and visualize their module, their cluster tree, and right underneath I show, of course, the color labels. So because I started with 3,600 genes, the function split them into 2 blocks. The first block contains 2,000 genes, the second 1,600 genes, and so you would now have 2 dendrograms. In practice, meaning in publications, we often don't even show the dendrogram. And why is that? Because our focus is on module eigengenes, and so we don't even bother to show that. In any event, let's now come to a third version of the analysis. So remember, we started out with what I call the lazy analysis. One R command does everything. Then I did the blockwise analysis, but now we come to the third analysis, which we call the manual or stepwise detection, which gives you as the data analyst maximum control over individual steps. And so remember, WGCNA starts out with defining the adjacency matrix, and then we turn it into the topologic overlap matrix and then into a distance, and then we carry out hierarchical clustering and then do dynamic branch cutting. And so I briefly go through these steps. So to define the adjacency matrix, we of course use again the R command adjacency. And if you wanted to get a signed network, you would now say, well, network, the type of it is signed, and here you specify the power. Remember, the default choice for the default choice of the power for a signed network is 12. In any event, let's proceed with this, and this will take about 2 seconds, and we have an adjacency matrix. Now, to turn an adjacency matrix into a dissimilarity based on the topologic overlap, we use one R command called tomdist. And this is now a dissimilarity measure, and we use that dissimilarity as input of FlashClust for creating a gene tree. Now, once we have a tree, we carry out dynamic branch cutting, and that is accomplished with the R command cutTreeDynamic, which implements 2 branch cutting methods. Remember, we often prefer the hybrid method, which is a hybrid between hierarchical clustering and partitioning around medoid clustering. And the great advantage of the hybrid method is that it leads to more robust module assignments because it inherits this property from partitioning around medoids clustering. Now, in order to apply the hybrid method, you also need to input the distance measure you used. So you, in this case, the cutTreeDynamic function takes as input the gene tree resulting from hierarchical clustering and also the dissimilarity measure. Now, as Peter mentioned, there is an important parameter that governs how the branch cutting is carried out, which is the deep split parameter. Deep split 1 results in larger modules. Deep split 2 or 3 results in smaller branches, smaller modules. All right, so we apply it, and after we apply this function, we get now module labels. Again, these are integers, and in order to reshuffle the labels so that they align to our previous module assignments, we use again the function matchLabels. And then we get these matched labels, and we assign colors to them using labels to colors. We have now the module labels we did, but remember, the focus of the analysis is to arrive at module eigengenes. And so to calculate module eigengenes, we use the R function called module eigengenes, which takes as input the gene expression data, and it takes as input the module assignment, your favorite module assignment. Now here, of course, we take the module assignment that resulted from the cluster tree analysis. But as a digression, let me mention that let's say you have a module assignment that doesn't come from a cluster tree. Let's say these are pathways from some database or gene sets. Then, of course, you could input these as pseudo module labels, right? Let's say you take KEGG pathways and you label them by integers and input that. So you could input anything and you get as a result module eigengenes. Remember, the module eigengenes explain the maximum proportion of the variance of a module. Now this function also, and so to get these eigengenes, you use again $sign eigengenes. The output of this function module eigengenes is a list, and you say, give me the list component that contains these eigengenes. But this output contains other list components that report, for example, the proportion of variance explained of each of these eigengenes. And also, it allows you to calculate the second component of eigengenes and so on. In any event, we arrive at these module eigengenes, and now what we do is we cluster the eigengenes. You know, that's an exciting analysis actually, because you can form clusters of clusters, right? Modules are clusters, and so you can go a step further. But in any event, this analysis serves the purpose of deciding whether modules should be merged. And so, just to explain the figure to you, so of course here's the hierarchical cluster tree where all the leaves correspond to module eigengenes, but right underneath it, we show the corresponding correlations. And so what do we learn? The purple module and the dark red module eigengenes are highly correlated with each other. Similarly, this group of modules is fairly highly correlated. And so, of course, it's now tempting to merge some of these modules. For example, these two modules should probably be merged. And remember how we have a parameter called merging threshold. So the merging threshold here could be chosen as 0.25, we chose that earlier, which indicates if these two leaves merge at a height of 0.25 or less, they will be merged. You know. So to merge closely related modules, we use the R function merge close modules. And again, you indicate here what is your merging threshold. How do you choose it? It really depends on the sample size. If you have a small data set, I want to say 30 samples, you would want a merging threshold that is higher, think like 0.25. Whereas if you have a data set with 500 samples, you get away with a lower merging threshold, maybe 0.15. All right. Now, after you apply this function, you then arrive at a new set of module eigengenes that corresponds to these the merged modules. And we can, of course, now visualize everything we've done. So this is the original cluster tree, and here I show the different module color assignments. So the last analysis was this manual analysis, and these are the resulting colors. Remember, we started out with the automatic analysis that did everything in one R command, and here are the corresponding colors. And so what do we see? We see actually very good agreement between the manual analysis and the one-stop shop analysis. The third color band shows the results from the blockwise analysis. And again, we see very good agreement of the module assignment. Interestingly, the blue module is somehow merged with the brown module, you know. And the truth is that there are good scientific reasons that the blue and the brown modules should have been called one big module, you know. Actually, it turns out that this module corresponds to macrophage related genes, you know. So there's a biologic story here that says this body weight related module relates to macrophage infiltration. But at the time when we published it, we didn't recognize that, and we kept it separate, you know. But interestingly, the two-block analysis puts them together, you know. So let's move on. Oh, I should mention that this, of course, is a highly complex cluster tree, meaning many of the modules are not very distinct, and hopefully you are luckier in your data that the branches are more distinct, and so you don't have to go through this pain, of having to decide should they be merged or not. Statistics can do so much. For example, here we merge modules if their eigengenes are highly correlated, but also, of course, you could look at the gene ontology enrichment results and say, well, there's evidence that these modules should be merged. And then you can easily accomplish the merging using these tools we described. In any event, let me move on and now relate the modules to the clinical traits. And what I'm after is to create this plot. I will zoom into it a little bit. So this is a plot that very often is reported as main figures in a scientific journal. So what does it show? So the rows here are, of course, the module eigengenes, and the columns correspond to these different physiologic traits. Each entry in this table reports, for example, the Pearson correlation between the module eigengene and the corresponding physiologic trait, and right underneath it is the corresponding p-value in brackets. I hope you can you see these numbers? So there's a sign, and in brackets is the p-value. And what do we learn? So the blue module is highly positively correlated with mouse body weight, also with abdominal fat, total fat, and so on. Remember that red indicates positive relations and green indicates negative relations. So for example, this blue module has a negative correlation with glucose insulin. And so this type of plot then provides an overview of your entire data set. It summarizes the expression data and for the rows and the physiologic traits. Yes? Yes, so in order to accomplish these correlations, it's really critical to have the trait measurements on the very same mice that were used for gene expression profiling. I think I didn't highlight that enough, So it's of course very straightforward to correlate a module eigengene, which takes a value for every mouse, with body weight, which is also a value for every mouse, so you just correlate it. Yes? Yes. Yeah. So let's assume you have categorical variable that takes only two values, 1 or 2. For example, it could be disease status. Then it's easy. You turn it into a binary numeric variable. But if you have a truly categorical variable with, for example, ethnicity, then one trick would be to turn it into a set of multiple binary variables. And of course, in case of ethnicity, you could have an indicator: Asian, yes, no; Caucasian, yes, no; Hispanic, yes, no, and so on. But finally, I mentioned you can also create what we call verbose bar plot, or a bar plot where the y-axis shows the mean module eigengene value, and the different bars correspond to different categories, So that would be the other way to do it. It's really ANOVA, where the dependent variable is the module eigengene. Yeah. Yes? So the question is, I guess, where does this discrepancy come from? And yes. Oh, how do I decide the merging threshold? Remember how we always tell you that we actually have very good default values. And in many analyses, I really go just with the default values. But this is now a network course, and I want to give you the nuts and bolts. So the default value for the merging threshold is 0.2. That works very well, which is a reasonable value if you are very familiar with values for correlations. If you have two module eigengenes that have a correlation of 0.2, they are really highly correlated with each other. And so if you have noisy data and a limited sample size, then one could argue the two of them are very similar. And to play it safe, I merge the modules. If we had a very large data set that allowed us to estimate correlations very precisely, let's say 1,000 arrays, and then I would say, well, if two module eigengenes only have a correlation of 0.8 together, I really feel not comfortable merging them because they may really correspond to different modules. And so it's a judgment call that is in part informed by your sample size and the amount of noise in your data, But if you expect if you think there's noise in your data, then it's often better to err on the side of caution and to arrive at larger modules, right? Because if you report, let's say, 50 modules, and you make a claim that there are really these distinct patterns in your data, and if you don't, and so it's a more bold statement than to say overall in my data set I see these 10 different themes, these broad patterns. So if you therefore it's often prudent to err on the side of larger modules and fewer modules. Yeah. For getting this tree. Oh yeah, okay. So let's discuss this. So in order to merge or to consider merging modules, what I need to do is I use, of course, the R function merge close modules, and I input the gene expression data. Further, I input the initial module assignment that I found. And the third thing is I specify the parameter cut height, which is really this height cutoff, So think of it again as branch cutting. I say my cut height here is 0.25, and so anything that lies below the cut height of 0.25 gets grouped into, you call it a meta module, a module of modules. And so these two things then get grouped into a meta module, and its color reflects one of these colors. It gets a new color. Yeah. And this new color that results can be accessed using, again, the dollar sign. So these are now the new colors. And but also the merge function automatically calculates new module eigengenes corresponding to these colors. Okay. So moving along, so I have now these module and trait relationships, but of course the next thing is to find exciting genes, for example in the blue module. And one way to do it is to look at module membership, also known as KME, the module eigengene-based connectivity. To do that, I use the R command signed KME. Why? Because it contains the sign information. Remember, KME is defined as the correlation between expression and module eigengenes. So applying signed KME to the expression data and these module eigengenes results in a data frame of KME values. And now I can plot the KME values in the blue module versus the gene significance measure I have for body weight. And so what do I learn? I should say many people focus on this reported correlation, and it's really misleading. I never do, but what do I look at? I look at the y-axis. So what do I learn? I learn that genes that have high positive module membership tend to be highly positively correlated with mouse body weight, whereas genes with negative values in that module, they have negative relations with mouse body weight. So the bottom line is this module, of course, relates strongly to mouse body weight, and I can now select genes either according to their association with mouse body weight, meaning high positive GS weight or high negative GS weight, or I could say select genes with high KME, the hub genes. Incidentally, you can do this exercise for other modules, and notice there are some modules where you actually see a negative relationship. But so look at the black module. Genes that are in that module tend to have weak relationships with mouse body weight. There are only a few. Certainly, these hub genes with KME close to 1, they have only moderate but negative relationships with mouse body weight. Now, the next logical step will be to output these KME values. Why? Because you would now select the 500 genes in each module and subject them to a gene ontology enrichment analysis. I do mention that the WGCNA R package has an R command called go enrichment, and you could just literally input genes there and you get go enrichment. However, I do recommend that you output these KME values and then use, for example, David or Ease, the software, or Ingenuity or WebGestalt, or we heard, of course, about the wonderful functionality of Cytoscape and pathway enrichment analysis from Gary Bader. And so you could do that, yeah? Yes? Yeah, so that's my favorite ad hoc number. So often people who are familiar with Go enrichment analysis know that you need a fair number of genes when you study Go enrichment. In the past, I sometimes used 200 genes, but why not 500, And so remember, the module sizes vary greatly. There's a very large blue module. There are other modules that are small. But I'm often too lazy to choose for each module a different number, and I want to get the quick overview, and therefore I sort genes according to their KME values, and I take the top 500 genes for every module and just look at them. Of course, when you write a scientific publication, then you start to become more careful, and you would want to think about how many you choose. All right, now what I will do is I skip a whole section in the tutorial and I move to ways of visualizing this network. Remember, I told you there's what is known as topologic overlap matrix plot, and you just use the R function tomplot to do that. But alternatively, you could also create a classical multidimensional scaling plot, and you can accomplish that by using the CMD scale command in the R. Also, you can use external software packages, for example, Visant or Cytoscape. And so to interface with these packages, you can, for example, use the R command export network to Visant. And so here I export the 30 top intramodular hubs in the blue module and then create an input file for Visant, and then Visant arrived at this type of plot. But similarly, we have commands for exporting a network to Cytoscape. Let me use the last few minutes to describe how we do module preservation analysis. So assume I want to verify which of the female modules can also be found in the male liver data. So to do that, I read in the male liver data, and ultimately I will use the R function module preservation, because it does the module preservation analysis. But notice that this function requires that you input what we call a multi-expression data set. And so the multi-expression data set is defined here. It's really a list that has two components. The first component are the female liver expression data, and the second list component are the male liver expression data. Further, remember that we need to input the module colors, for example, found in the female reference network. And so that is again a list. And so then we run this module preservation function, and one critical parameter is the number of permutations. And because this is a tutorial, I only indicate 10 permutations, but for a scientific publication, you would want to run 200 permutations. And also, here I specify that the first list component, meaning the female liver data, are the reference network. And so I skip through a lot of the output and just show you the type of figure that we get. And so notice here I show the Z summary statistic, which measures module preservation in the male liver data, and the x-axis shows the module size. And what do I learn? Well, most modules are highly preserved, so most female modules are highly preserved in male liver, which is reassuring. I do notice, of course, there's a relationship between module size and preservation. And remember, I mentioned to you, this is a feature, it's not a bug. If a module is large and its connectivity pattern is preserved, then you have far stronger statistical evidence than if you have a small module and its connectivity pattern is preserved. Now, the one thing you should notice, there's one module, the light green module, that falls below the threshold of preservation. It's not preserved. And so we could now be very tempted to write a very high-profile articles where we say there's this sexual dimorphism. We have one module in female livers that is not preserved in male livers, which could explain all sorts of reasons why certain features also show differences between the genders. For example, body weight, or fat distribution, and so on. But before we write this high-impact paper, let's do one thing, and let's look at the heat map of that module, which, by the way, you should always do. You should always look at heat maps of your modules, because they could highlight data quality issues. And this is exactly the case here. So here we see the light green module, the genes are rows, and the columns correspond to these female liver samples. And you notice this one green line, which corresponds to one mouse where all the genes are highly underexpressed. That seems to be a technical artifact. It could be some tissue contamination. It could be there could be any range of reasons why there is this outlying mouse. But remember, earlier I removed already an outlying mouse, but I didn't catch this mouse. And so in hindsight, what we learn is that the reason why this module was not preserved in male livers is because this module is due to an artifact, and therefore we couldn't find it. Remember that we mentioned that a very valuable tool for aggregating multiple networks is consensus module analysis. And so let's say I want to find modules that by definition are present both in the female and male liver data. So in order to accomplish that type of analysis, I use an R function called blockwise consensus modules. It's analogous to my earlier function blockwise modules, but now it finds consensus modules. And the only difference is that we now input a multi-expression object. Remember, that's a list whose first component are the female data and the second are the male data. But all the other parameters stay in place. And then we get as output module labels. These are now the consensus modules. And we get also a list of module eigengenes. And so then I can visualize the cluster tree, and these are now this is a cluster tree whose branches correspond to consensus modules. And I can again relate these modules to the physiologic traits, but now I need to distinguish between female and male livers. So these modules correspond to consensus modules, but now I show their correlations with the physiologic traits in the female data. And analogously, I would get a second table that shows the results in the male data. With that, I stop here. And I want to remind you that we have a reception over here. So I hope to meet many of you. But so don't run away. But do you have any questions at this point? Yeah, please. Yeah. So when I created the dendrogram for the blockwise consensus analysis, I chose a maximum block size of 5,000 so to ensure that it's a single block analysis. You know. Yes, over here, please. Yeah, I didn't quite hear you, but did you say whether I can use it for count data? Or? Yes, so definitely. So, yeah. Let me already mention that often, of course, RNA-seq data are count data, and people just apply these same tools to RNA-seq data. But more generally, you sometimes have real count data, for example, from microbiome studies or or peptide count data. And we and others have certainly applied these methods without much ado to just count data, you know. And the reason is the correlation is really a reasonable measure for correlating or relating count data. There's really no reason not to use it, you know, and it works very well. I'm sure there are some statisticians that will now revolt at this statement because they have their Poisson regression tool, for example, to relate count data and so on. But the truth is that these data contain so much noise that these subtleties of the dependence measure are really dwarfed by the noise in the data. And the correlation is a wonderful tool for relating numeric variables, including count data. And we have applied it to peptide count data, and it worked very well to reproduce known biology. So I feel confident of making this statement. Yeah. Other questions? Yes. 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 So the question is, when we apply these methods to DNA methylation data, for example, measured on the Illumina 450K platform, then there are some valuable pre-processing steps, you know. So just to tell you, I certainly applied these methods to what they call beta values. You can apply them to these M values. I found no difference. I used the beta values. But then there, on the 450K platform, there are two types of probes, the type 1 and the type 2 probes. And they're wonderful pre-processing approaches to put these probes on the same footing. There's BMIQ is a recent method, and SWAN and so on. I use these tools, I find they add value, so I think it helps you. I should perhaps highlight that we always assume that the user used proper normalization steps, so you as the data analyst will know what is the most widely accepted and best way of pre-processing your data, and so do that, and only then apply higher level tools such as WGCNA or any other of these methods. Yeah, I have recently published a paper where I analyzed multiple methylation data sets from different human tissues, and I used the blockwise consensus module approach to find modules that consistently relate to age, irrespective of tissue type. And so that worked quite well. So the consensus network analysis is really a wonderful tool for aggregating multiple data sets. And it's a very conservative approach because it really conditions intrinsically on every data set, And so, it allows you to find the low-hanging fruits, Yes. Yeah, for the DNA methylation data, I really use probe-level data, the CpGs, Why? Because I can, The blockwise module detection analysis allows me to analyze hundreds of thousands of variables. Having said this, if you have a good rationale why you want to aggregate multiple probes per gene, that by all means go ahead. Again, the pre-processing decisions should be guided by the purpose of the analysis. Yes? Oh, no question. All right. Thank you all for coming today.